Okay. All right, so today's lab is the RBCs going into all these different solutions. And hopefully like this is gonna kind of make it more real about, why you need to think about osmolarity, how osmolarity is related to concentration, and also how tonicity depends on what kind of solute and all that. So, and also you can see why this process of getting your objectives and hypotheses is usually iterative. If you turn something in and I sent back some comments about like, this needs blah, 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 that's normal. That's normally how this works. It's quite rare that I just say like, whoa, bingo, you just totally nailed it. You're good to go. So it usually is an iterative process. That being said, you need to engage in that iterative process. There's, again, most of you are. For those that turn things in last moment, I don't have time to like give you feedback. Or if, so make sure that you make sure you also take the initiative. Like some people turn some stuff in that was, what do they call it, kind of phoning it in. It's like, you didn't even try. You didn't even go watch that video or go back and listen to what I was saying in the discussion about how to think about your hypotheses or the objectives. Everything in your objective is laid out very explicitly in a couple of different places. So make sure you take the initiative to like kind of engage the material. That being said, let's do
this. Dr. Eager, can I ask a quick question about the variables for this lab?
Yeah, for sure.
So is it going to be how the red blood cell reacts, or is it what we see in the supernatant, like after the centrifuge?
So it's a mix of both.
Yeah.
The initial dependent variable is going to be whether or not the supernatant is red, which is evidence of hemolysis. That is the most clear thing that we can look at. When we talk about the actual data and actually see pictures of the test tubes, two things. One is crenation, you can't tell for sure macroscopically, but you can get a hint. I think I drew on the board in that little intro thing about if you have raisins, like versus.
Yeah, they shrink.
Right, they're going to take less room. So you can use that to like get evidence of crenation. Like if you have, you started with 6 drops of blood in both tubes, but one pellet is smaller than the other. That's evidence that there's some crenation going on probably.
Okay.
And then we're also going to, or we would look at things under the microscope. And there you have more evidence. Did something actually, because normally just from after you centrifuge, you can't tell definitively that it's crenation versus just isotonic.
Right? It's just intact.
It's just intact. Yeah. So, however, under the microscope, it's more obvious, like the cells look like little puckered asterisks versus look like blood cells.
Okay, thank you.
So that's a good question, though. Again, the main thing, the first thing we're going to be able to tell is, did hemolysis occur? And again, what is the evidence that hemolysis occurred?
Like a red, pink centrifuge, not a pellet at the bottom.
Exactly. There'll be a red supernatant, though the solution will stay red because the hemoglobin has spilled out and is now dissolved in the water. And and the pellet can be more or less, like if everything is completely exploded, there's not much of a pellet at all. If some of them exploded, but not all of them, maybe you'll see some pellet, but not much of a pellet, right? So, okay, so let's briefly review. Actually, before we go into that, I want to go over the chemistry homework because I graded the chemistry homework. And some people did awesome and some people did not awesome. So I'm going to just go over it. If you are one of the ones who did not awesome, again, these are key concepts that we're going to keep using. Like next class, we're going to talk about how does a ligand bind a receptor? How come you trip when you eat magic mushrooms? Like, what is it about the psilocybin molecule that actually can bind and activate the serotonin receptor? It's going to be all about hydrogen bonding and ionic bonding, and it's going to be about protein structure with alpha helices going through the membrane, and all this stuff we're talking about is at the core of these, physiology. Even though you're like, I'm done with chemistry, I want to hear about like how cells work. It doesn't make sense unless you understand the chemistry. So, why an exogenous ligand actually can bind the receptor has everything to do with the chemistry. So let's take a look. I'm going to bring up the chemistry homework. Hold on 1 minute. Module. Okay, I can go share screen. Chemistry review homework. All right, so most people wrote it on there. I'm not quite sure. So yeah, no, let's just say, here we go. Oh, I want to open it. I can do it that way. Okay, so here we have it. Pretty much everybody got this one right. What kind of chemical bond binds NaCl? Let me do my plus here. So what kind of chemical bond binds NaCl?
Ionic.
Or ionic bonds. Whenever you have one of these alkali metals and a halogen, something from the first and second to last column of the periodic table, it's pretty much going to be an ionic bond. And again, it's critical that if something is bonded ionically, because that's going to make a huge difference in the relationship between molarity and osmolarity. Whenever, in our experiment today, if you have a solution of NaCl, you know that to get the molarity, you're going to have to just multiply times 2. On the other hand, and other things, KCl, calcium chloride, lots of things. What kind of chemical bond binds the atoms of sugar? This is just glucose.
Covalent.
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 Professor, again, at least on my screen, I see the corner of your web page.
Nabbit, I see what happened. Okay, hold on 1 sec. We are going to. That's because I moved the screen. Yeah, I know what happened. Here we go. Bing. All right. So covalent bonds, and what happens to covalently bonded molecules when you put them in water and look at their osmolarity?
They do not dissociate.
They don't dissociate, so they stay intact. So any kind of carbohydrate, like say sucrose, or pretty much everything that was not NaCl in our not NaCl or ammonium chloride in our experiment, is going to be covalent and is not going to dissociate. So molarity and osmolarity are the same. There were quite a few people who got fancy, like splitting up urea and glycerol and sugar into lots of pieces and got, large osmolarities and stuff, but that's not going to happen. When you have something that's covalently bonded, one solute molecule is just one particle. But again, that's why it is so important to be able to distinguish between, is that an ionically bonded thing or a covalently bonded thing? Because it makes a huge difference in osmolarity, which makes a huge difference in, as we're going to see, whether cells are happy or shrivel or explode. What kind of chemical bonds attract one molecule of water to another?
Hydrogen bonds.
Hydrogen bonds.
Hydrogen bonds. And here, this one, a number of people, a lot of people, I think it must be in some book, you know, it says another place you find them is in ammonia, you know, NH3. But if you just wrote that, I only gave you partial credit, because I don't know if you're meaning, you know, within the molecule of ammonia or between separate. So it's between separate molecules of ammonia that are holding themselves together. It's, you know, the actual single ammonia molecule is held together by covalent bonds. So I only gave you full credit if you wrote, you know, holding, or if you know, two different molecules holding together or things like that. pH. You know, I took a lot of times people like, what does pH measure of? What does it mean, alkaline or acid? Some people like went circular. You know, acid means the pH is like more than, you know, or less than 7, but like, what does it mean? Like at the core of it, what are we actually measuring?
Hydrogen ions.
The hydrogen ion concentration. And then I wanted you to be able to say, like, more acidic is, you know, getting more higher hydrogen ion concentration compared to more alkaline is less. Moles, again, most people got it, but some people still don't know what a mole is, which you probably want to know what moles are eventually. Again, think about like numbers of eggs in a carton. It's like number of atoms or molecules in a handful. It's like 6 with 23 zeros after it, atoms or molecules of something. And then molarity is just going to be moles per liter. Some people got weird with getting into molality, like, you know, moles per kilogram of solute and things like that. You know, that's something else. We're not going to deal with that in this class. This is just straight up mole solute per liter solution. Osmolarity. And what is osmolarity?
Is the solute, the concentration of the solute in a solution that affects osmotic pressure?
Yeah, so it's the sum total of all the solute particles that are going to affect. When we talk about molarity, we're talking about some particular compound. When we talk about osmolarity, we just mean all the little particles that can affect the osmolarity or affect osmosis. You know, so, and is it always the same as molarity?
No.
No, in particular, when is it not?
If the molecule dissociates,
Molecule
it's
dissociation is going to be higher. Percent solution, you know, percent solution, at the core of it, I just wanted someone to say, like, you know, grams of solute over grams of solution times 100. If it's by weight, or, you know, volume, mils of solute over mils of solution times 100. That's at the core of it. Is just kind of by weight or by, it's a way of specifying concentration by weight or by volume. Equivalence, most people got like 1 mole of charge, positive or negative. Oh, this one, people got weird or got circular on here. What determines whether a particular molecule is hydrophobic or hydrophilic? Yeah, what is the core thing you need to look at? What property of a molecule will help you determine whether it's going to dissolve in water or not?
Polarity.
Polar, non-polar.
polarity.
Polarity. Some people kind of spun it around. It's like, if it dissolves in water, then by definition, we're going to call it a polar molecule. It's like, no. It's like, it's probably a polar molecule, but polar has to do with local charge imbalances and stuff, or charge imbalances in general. So you need to go from polar will be hydrophilic, non-polar will be hydrophobic.
Okay.
What is an enzyme? Again, most people got this, the catalysts. And they're so important because most of the reactions in the body aren't going to run on their own, or they'll run way too slow. And the other things that we can use to speed up reactions, like put things at really high temperatures, for instance, is not practical. So we use enzymes to make the reactions go. This part, some people did great and some people like really didn't. So let's talk just a little bit about. So the first part here, if I'm asking what is the molarity of physiologically isotonic glucose, there's a whole bunch of, actually a non-trivial number of people got 0 on this because they didn't even answer in molarity. They answered in osmolarity, which is not what I'm asking. The osmolarity is given. The answer needs to be in molarity of glucose. So when you're answering a question, make sure you read what is the question asking. And then again, physiological fluids using 300 milliosmolar as a starting point. So in all these cases, that's your starting point. And so 3 or 0.3 moles, and I'm writing with my mouse, so this looks like, and I apologize, the grading, I've been Googling, trying to figure out how can I use a stylus. Canvas is really not easy to grade in. So 0.3 moles particles per liter. And molarity would be, I want moles of glucose per liter. So it's really straightforward. I just need to convert, I need moles of glucose on the top. So 1 mole of glucose is just 1 mole of particles because it doesn't dissociate. Again, pardon my writing. So it's 1 to 1. So that's just going to be 0.3 molar glucose, kind of by definition. If moles of particles will cancel out. So I'm left with moles of glucose per liter. So it's really straightforward. It should be 1 step. You could even use, you have to show your work. So you could use your words though. You could just say, because glucose does not dissociate, the molarity will be the same as the molarity. For saline, it's going to be slightly different. We'll start the same, but instead of having the conversion of 1 to 1, it's going to be 1 mole of NaCl is going to be 2 moles of particles because it dissociates. So again, the moles of particles will cancel, but now we have this division by 2, so it's going to be 0.15 molar NaCl. Right? And again, it's just 1 step. People got all funky on this one. Like, well, let's see, I know that the physiologically saline we said was 0.9%, and let's like go. It's like, what are you talking about? It's like, it says start here, and in 1 step, you get the answer. So, in fact, it was really kind of hilarious to me that a lot of people, I think, were using the stuff that I wrote on the board in lecture. So they actually wrote this, what was the right answer for this as the first part for question 14. And if they would have just written this up here, they would have got credit instead of getting a 0 on this. But I think that just means that they didn't quite understand what's going on. So hopefully in the future now, when you think about osmolarity and molarity, you'll realize it's just a simple relationship here. If you just divided by 2, but you didn't tell me why, I also only gave you half credit. If you said, because it dissociates into 2 particles, then I would give you full credit. Or if you showed me the conversion factor that explicitly mentions moles of NaCl to moles of particles, you got full credit. This one, most people got correct because all you had to do was write down what I wrote on the board. And if you go back to the, you know, if you were didn't have time or didn't have enough energy to go back, you know, that there were people who got 0 on this one. And then some people lost points because they got really sloppy and just wrote, oh, it's 9%. It's like 9 and 0.9 are really different. It's a factor of 10, right? If somebody weighs 100 pounds or someone weighs 1,000 pounds, that's really different. You can't say, it's almost there. It's, come on. It's like, it's so not almost there. It's totally different, right? Like 1,000 and 100 are really different. 0.9 and 9 are radically different. So you have to be careful. You have to worry about decimal points and keep track of that stuff. And then finally, speaking of moles, so this one, I put this here because part of what you're going to be doing, and in fact, what we're going to do in our lab next class is not going to be an actual lab. It's going to be talking about searching the literature. It's going to be about what are the tools and techniques you have for seeing what do we already know about stuff people have studied. So part of what we're going to be doing in lab is getting good at, you know, I'm curious, is fiber good to like prevent colon cancer? Like, how do I go? Where do I start? How do I look for it? Where's the answer gonna, most reliable answer gonna be? So I put this here just to get you realizing, like, you know, using your search tools and keywords and stuff. You know, I got a lot of answers here. A lot of people did get the right guy, Kim Philby, who was a very famous mole who was in World War II. Other people put other guys. Obviously, they were spies, they were double agents, but you need to do the next, remember that sanity check. If you go into Wikipedia and read about this person and it says that they were in the 70s, it's like, okay, they're not actually in World War II. Or if you read they actually were working for the Russians and double agent spying on the FBI, no, that doesn't make sense here. You know, so some people wrote down some interesting famous spies or double agent spies, but you needed to make sure that they were a senior officer in the British intelligence, double agent working for Russia, the Soviet Union, and that the time period was correct. World War II and the beginning of the Cold War. And then Kim Philby's the main dude who comes up. Who is this? This is from other semesters. I use other famous moles. Does anybody know who is the famous mole who feels that kind of the song of spring calling longingly to him? And what book this was? He goes like, oh, onion sauce, hang spring cleaning. And he goes off on an adventure with the otter and the rat and everybody. Did people ever read this book? It's Wind in the Willows. It's definitely worth reading. It's very sweet. All right. So are there any questions about the chemistry homework? You know, again, some people, in fact, just to give you a sense. Yeah, some people got perfect scores or more than 100% because you got an extra half a point if you got Kim Philby here. So it was possible to get 15.5. The scoring for this thing was 1 point for each of them, except you got 2 points for the osmolarity one because that had 2 parts. The osmolarity one, you got 1 point if you were you had moles particles per liter for the definition, and another point if you said no, it's not always the same. Sometimes it can be higher if the thing dissociates. So 15 points, 15.5 if you count the extra credit. And again, some people got 15 or 15.5. Some people got 9 or 10. If you are one of those, again, go back and make sure you got these concepts, sooner than later. Partly because you got an exam coming up where this is all coming back, but partly, more importantly, because just everything we're going to talk about is going to be confusing. Like we're going to, again, next class, we're going to talk about a molecule of serotonin or psilocybin binding to a serotonin receptor. And if you don't understand this stuff here, it's going to be like me just spewing at you. It's like, it's like, and the purple aliens ran around with purple hats and ran and then got into cars and flew into the sky. It's like, what are you talking about? It's going to be like that. Let's see. All right, stop share. So what time is it? It is 10:43. So for this lab, I would now like to just go through the objective and the hypotheses together before we actually dig into this. And then maybe we can take a little break and then we'll start the actual lab itself. Well, watching the lab itself. So, whoops. So who would like to share their objective for today's lab?
Me.
All right, Maria Portela.
Okay. What is the correlation between the osmolarity of the solution and the type of solute, I wrote penetrating or not penetrating, and the osmosis inward or outward the red blood cells?
Right, so she's got, there's 2 independent variables. One is the concentration of the solution that we're putting the cell into. And the other one is the type of solute, right? Because those are both going to be important, playing important roles. We have some types, and what is the main, when we think about the types of solute, it's going to affect 2 things. One, it's going to affect, because ultimately we're worried about osmolarity, right? So the type of solute is going to determine the relationship between the molarity and the osmolarity, which we care about, right? 0.3 molar NaCl and 0.3 molar sucrose. They both have the same molarity, but the osmolarity is really different and their effect on the cells is really different. So unless you're thinking about the type of solute, you are going to totally mess up when you're trying to figure out what's happening, even if you know the concentration of the solute. So we're going to be looking at both the concentration, but also type of solute. What other aspect of the solute type is going to affect our results?
Penetrating versus non-penetrating.
Exactly, penetrating versus non-penetrating. That makes a huge difference. You can have an isoosmotic solution, so here we're talking about molarity. Okay, now we've gone to osmolarity, but we can have 2 solutions of identical osmolarity, but have very different behavior of the cells when you put them in, because if you have a penetrating solute, as we've talked about, the solute actually is going to enter the cell and the water is going to follow, and you can still get out of balance and go into have a hypotonic solution, even though it's an isoosmotic solution. So you need to pay attention to the type of solute, both to calculate the osmolarity from the molarity, and also even once you know the osmolarity, so you can determine, is it actually isotonic or hypotonic? You know, just because you know the osmolarity doesn't mean you know if the cell is happy until you also know if it's penetrating or not. So objective, we're going to look at varying types of solutes as well as different concentrations of solutes. And then what is our dependent variable? What's the thing we're going to look at?
Hemolysis.
Yeah, we're going to look at what happens to the cells. So like Maria was saying, osmosis, which is basically what we are going to be looking at. And more specifically, we're also going to be saying, did the cells burst? Do they hemolyze? So, you know, or you could say like, you know, what is, you know, what's the effect of the concentration of the solution and the type of solute, you know, on the behavior of the red blood cells in there, specifically, do they hemolyze or things like that? There's different ways you can say it. But so is everybody's pretty clear on the real question we're asking? We're gonna get to play around with different solutions of varying concentrations, varying types of solutes, and then look at, you know, what happens to the RBC. You know, based on whether water comes in, water comes out, or there's an equilibrium. So either it's going to be happy, isotonic, it's going to swell and or burst, hypotonic, or it's going to shrivel, which is hypertonic solution. All right. That being said, test tubes. Number one, distilled water. This one cracks me up to see how creative people get with the osmolarity and everything with this test tube. All right, so if I have a test tube, if I have distilled water, what is the osmolarity of this one?
0.
0.
0. Because osmolarity is number of particles per liter of solution, and this is just pure solvent. There's no solute at all. There's nothing dissolved in there. That's by definition, distilled water is just the pure solvent without any solute at all. So this has 0 osmolarity. And then if we want to figure out which way does water move once we put the cell in, what do we have to do?
Determine if it's penetrating.
There's no solute to penetrate or not penetrate.
No, observe about how the osmosis, the diffusion of water into the red blood cells, what is happening to the red blood cell.
How do I predict whether or not osmosis is going to happen in or out or not at all?
Well, the concentration of the red blood cells is always going to be higher. So then we know that the H2O would go into the cell.
Right. So what
The
is
osmolarity of the red blood cell.
so right? So in every single test tube, I'm going to calculate the osmolarity, and then what am I going to do next? Once I have,
Compare it to the red blood cells osmolarity.
yeah, exactly. Which is what?
Is 0.3 milliosmoles.
Right. So every time we're going to compare whatever we get to 0.3 osmolar, which is the RBC. Right. So we always, the RBC is always going to be this. And then we're going to see, we're going to always calculate what's in the test tube. And then I compare them and I can say, oh, 0 is less than 0.3. So what do I call a solution if the osmolarity is lower than my red blood cell?
Hypotonic.
Hypoosmotic.
It's going to be hypoosmotic.
Okay, hyposmotic.
Hypoosmotic because it is less than,
Osmolarity, yes.
and then the last thing we can do, the tonicity, which way does the water go? If I have a cell in a hypoosmotic solution, which way does the water want to move?
Into the
cell.
cell.
The water always goes to where there's more solute. So this is going to be hypotonic.
Associate.
And water moves
Hypotonic.
in.
Okay.
When I was trying to learn, in fact, I remember, I probably shouldn't guess, probably like 40 years ago, I was trying to remember this, and I remember thinking like hypotonic distilled H2O, and then like big bloated and filled up and blowing up or something. I remember that was how I tried to connect all these words together, like distilled water, hypo, and blowing up and everything. But you want to, whenever you put a cell into distilled water, it's not a good thing for that cell. If you're a plant cell, it's okay, because a plant cell has a cell wall made out of cellulose. That is like it's basically like a girdle that really holds on tight. So it just pushes hard against this, cell wall, and that's what actually makes the thing have turgors. It's why if your plant actually doesn't have enough of that, it's going to wilt. So plants, you can put a plant cell in distilled water and it's going to be okay. But that's because it's got like a pressure jacket on that keeps it from exploding. It's like those guys in those flight suits or whatever that kind of push them in so they're going at the high G's and stuff. So for the distilled water tube 1, this should have been pretty straightforward. This should have been the most straightforward of them all. There's no solute, so 0 osmolarity, which is as less than physiological as you can get, which means as hypoosmotic and as hypotonic with a vengeance, basically. So test tube 1. If this doesn't,
I understood tonicity as only if a solute can't penetrate the cell.
No. Tonicity is water moving across. Hypotonic means water moves in. Hypertonic means water moves out. And isotonic means there's no net movement of water.
I understood that, but as I was looking at the definition of tonicity, it is only relevant if the solid particle is non-penetrating. Am I understanding this correctly?
No, that's not, because we're going to see we're going to see hypotonic solutions with penetrating solutes in today's lab.
Like urea.
Yeah, so hypotonic just means the water moves into the cell when you put the cell in the solution. And our solutions that have penetrating solutes are going to be hypotonic. So, yeah. All right. Test tube 1. Next we go to test tube 2. And actually these are all the NaCl ones. So we had 0.1, what is it? 0.05 molar, 0.1 molar, 0.15 molar, and 0.3 molar. So in all of these cases, how are we going to go to osmolarity?
Multiply by 2.
Just multiply times 2. So this is going to be 0.1 molar, osmolar, 0.2 osmolar, 0.3 osmolar, 0.6 osmolar. You just multiply times 2 because we know NaCl dissociates. So then if we think about these two here are going to be hypoosmotic, meaning their osmolarity is less than 0.3. This is going to be isoosmotic because the osmolarity is exactly 0.3. This is going to be hyperosmotic because it is greater than 0.3. Right, so this should all make sense. And then, is NaCl penetrating or not penetrating?
Not.
It does not penetrate. And so then we talked about if it's not a penetrating solute, then the relationship between tonicity and osmolarity is really easy. It's just hypoosmotic is hypotonic. Isoosmotic is isotonic. Hyperosmotic is hypertonic. So what's the water going to do in these cases? Hypoosmotic?
It's going to go into the cell.
Going to go into the cell, and minimally it's going to inflate. If it inflates with enough pressure, it will pop and hemolyze. If it's an isoosmotic solution, which means isotonic, what's going to happen to the cells?
It will shrivel.
No change.
No, nothing. Nothing. Nothing will happen.
No net movement of water in and out. They should look happy. Under the microscope, they look kind of like that. What about in a hypertonic solution? The hyperosmotic becomes hypertonic.
Shrivel.
It loses water, and that's where you end up getting crenated cells. And so this all should make sense pretty much. And then, what I do? A 6 I did with sucrose. Again, this one, what is the osmolarity of 0.3 molar sucrose?
0.3.
It's going to be 0.3 osmolar because it doesn't dissociate. Is sucrose penetrating or not penetrating?
Not penetrating.
Right, it doesn't penetrate. It's just a covalently, it's a big polar molecule, large polar molecule. So it's isoosmotic. Whoops. Isoosmotic, isotonic because it does not penetrate. So what's going to happen to the cells in there?
No change.
No change. This should look pretty much exactly the same as the test tube with the 0.15 sodium chloride, right? It's just a isotonic sucrose solution instead of an isotonic saline solution. Test tube 7, 0.3 molar urea. What is the osmolarity of it?
0.3 molar.
It's going to be 0.3 osmolar.
Or smaller.
Again, because urea is just a small covalently bonded molecule that doesn't come apart. But what about, is it penetrating or non-penetrating? And this you know because I've been telling you a bunch.
Penetrating.
This is penetrating. Back. So that's going to come up, be important. So 0.3 osmolar, which means it's isoosmotic, to the blood cell, the same osmolarity as my blood cell, 0.3 inside, 0.3 outside, but it's a penetrating solute. Again, so when you put the cell into this solution, it's going to enter into the cell. And as it enters into the cell, it's going to start increasing the amount of dissolved stuff in the cell, and water follows the solute. As the solute increases inside, water is going to come in and follow along, which is going to blow this thing up. So that's why it's going to be hypotonic, even though it's isoosmotic. Right? So does that make sense?
Yes, one question, Professor, because I wrote this. Inside the red blood cell, when urea penetrates, the osmolarity will increase?
Yeah, the molarity inside is going to increase, so it's no longer 0.3, and that's why now the balance is off.
The water diffuses. Okay, thank you.
So the water will diffuse in and follow the urea.
Okay.
Test tube 8. All right. So this is the ammonium chloride. So this one is a interesting one. So we have 0.15 molar ammonium chloride. What is the molarity of this thing?
0.3.
It's 0.3. So this is going to dissociate. It's going to become ammonium ion. So it's going to be at 0.3 molar. I mean, not molar, osmolar. So it's isoosmotic. What about the tonicity? What's going to happen to the cells when we put this in there?
Isotonic?
So if all else being equal, that's what I would guess. I would guess it's isotonic. Because these are charged things that aren't going to cross the membrane. As we're going to find out, that's probably not the case. In fact, yeah, we're going to talk more detail about what the results are here after we get the results. But I would say, if I was just coming up with my hypothesis, it would make sense to think it's isotonic. What really happens with it is actually really complicated. It's only recently that I've even fully understood myself why what happens happens. But we will go into it in a little while when we're discussing things a little later. I'll give you a hint though, it has to do with antiports in the membrane. Turns out red blood cells have this thing that exchanges chlorine ions for bicarbonate ions. That's going to be playing a role in how this affects the red blood cells. So, and then finally, test tube 9, glycerol. You know, at 0.3 molar does not dissociate, so it's 0.3 osmolar. So isoosmotic. What's it going to be like in terms of tonicity?
Hypotonic.
No, it's penetrating, so it'd be hypotonic.
Right, so penetrating, so it's a little tricky. So glycerol is kind of a mixed kind of thing. It's not purely nonpolar. It's got like those hydroxyls hanging off it, but it's also got a side that's just like kind of hydrocarbons. So glycerol is one of these things. It's an alcohol that it can kind of have a foot in both worlds. It can interact in polar stuff. In fact, I mean, they stay together. It stays together as a liquid because of hydrogen bonds between it itself. But then it also has the ability to interact with nonpolar things because it's got a whole nonpolar side to it as well. So exactly what it's going to do is if it does have enough permeability, goes in tight, Maria. If it has enough of a permeability to get through the membrane to upset the balance, then what would happen?
Be hypotonic.
It would be hypotonic.
Mhm.
It can get through, but not so well. So by the time we actually spin it down, it hasn't had a chance to actually make it into the cell. What will we see?
It will stay isotonic.
It'll stay isotonic. So here it matters actually. If we're looking at like a person or a cow. It turns out that the permeability of red blood cells to glycerol is different in different mammals. There's actually a paper that just explores glycerol permeability in different mammalian cells. It turns out human cells are quite permeable to glycerol. So if you put in fact, this was kind of an interesting thing for me. Like when we used to just do this lab with human blood, you know, whenever we spun it down, the glycerol would get in the cell, the cells would explode. So in my world, it's like glycerol is very permeable into red blood cells. It's a no-brainer. And then just a year ago, we switched over and started using bovine blood when we were having trouble getting the nurse to take our blood. And the cells didn't explode. The cells were like, what the heck? How come human cells explode and cow cells don't explode? And that's when I had to do more research on relative permeabilities of red blood cells in different mammals to glycerol. And it turns out cow blood is much less permeable. So if I wanted the cells to explode, the cow cells to explode, what would I have to do? Assuming it's permeable, but much less permeable.
Increase the concentration of glycerol.
I could increase the concentration of glycerol. What would be another way?
Raise the temperature.
Raise the temperature, or I could just wait longer. You know, it's getting in, but it's just it's having a harder time getting in. So one of the things that we did this summer for you that's not part of the original protocol is we kept one of the cow bloods with glycerol and let it incubate for a half an hour before we spun it down to see the difference between just mix it up and spin it down versus give the glycerol a chance. And so that's something you'll get to see when we look at our results today. So are there any questions? We just kind of laid this all out. Is everybody good to go?
So we're using human blood in this experiment or bovine blood?
Bovine blood.
So all this, sorry, Professor, will be sources of variability, like the type of blood, if we're using cows or human.
The amount, in the case of the glycerol, it actually turns out how long the blood is sitting in the solution before you spin it down makes a huge difference for the glycerol. Because,
okay.
you know, so the cow, it is of bovine ilk. One end is moo, the other milk. That was like James Thurber. No, Ogden Nash. All right, we are now going to go into the actual doing the lab. Are there any other questions about any of this before we get into the actual process? Okay. And it's been, are people feeling like you can sit, it's pretty passive. Watching the YouTube is passive. So I think we can just go into it right now. So I'm going to share the screen. We're going to go to the module. Ah, come on. Okay, the first one is going to be just you watching me go through the process so you can see how we actually set it up. There's nothing for you to record really. It's more so you can see the process. All right, so I'm going to start 3 mils of distilled water in test tube 1. Again, we've already discussed the proper use of these pipettes. I've sucked up to the 0 here, putting it in the appropriate test tube. Now I dispense down to the 1 mils dispensed, 2 mils dispensed, 3 mils dispensed, and that's that. I'm going to get 3 mils of my 0.05 molar NaCl. Again, just suck up to the 0 and put that in the appropriate test tube. Dispense 1, 2, 3 mils. Notice it's going to be important for you to be precise with the volumes. One, just because we want to make sure all the conditions are, controlled and as similar as possible, except for our dependent variables, which we're changing. But also because we need to make sure things are balanced in the centrifuge. And think about like if your washing machine or your dryer is off balance, there's more weight on one side or another, it goes boom, 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 boom. It's even worse if you're going super, super fast, high-speed centrifuge, and we don't want any disasters. And you need to make sure Tina will talk more about the balancing, but part of the balancing is making sure each of your tubes is got the same volume. So what I'm going to do is I'm going to put 0.3 mils of each of the appropriate solutions into their appropriate test tubes, and then we will come back in a moment. Good. All right, so we have each of the test tubes with the appropriate solution. And now each test tube is going to get 6 drops of the blood. Like I said, make sure it is well mixed because this is. All right, so what of the different kinds of mixtures we talked about, what would we talk about blood cells and plasma? What would you call that?
Suspension.
That's a suspension. So if it's just been sitting around, all the blood cells are sitting on the bottom. So you got to make sure you shake this thing up. Otherwise, you're just going to get mostly plasma and not get the cells. Suspension, you're going to have the cells falling out. And because we want to only vary, the solutions, we want to make sure the amount of blood in each test tube is the same. So these drops, we try to make them as consistent as possible. It's going to be 6 drops. And it's not just like, around 6. I really want to do 6 well-formed drops. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. I should also mention, this is cow blood, so we don't worry about the safety as much. But if this was human blood, obviously we would be very concerned about safety and be using universal protocol, universal precautions and all that. Let's see. I should also mention blood, once it's out of the body, starts clotting. We're going to see that in much more detail when we do the blood lab later. In order to make sure the blood stays liquid like this, you have to use some kind of anticoagulant. There's a whole bunch of different things like heparin and things that you add to keep the blood from clotting. But normally, like if you like go to the, you can go to the store and just, there's different soups and stuff that are just made with pig blood and stuff. And you can just go to like a Filipino market and just get a little vat filled with pig blood or something for your soup. And if you open it up, it's going to be like Jello because all of the blood has kind of clotted and everything is all, so if you just go to the market thinking, I'm going to get some animal blood and recreate this, it's probably not going to be liquid like this. You need to treat the blood right after, as soon as you get it, so it doesn't turn into a big blob. All right, I'm going to continue here.
Another 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Notice I'm also refilling before it's empty so I don't get into a weird position where I've done part of the drops and get foam coming out and then it's not a well-formed drop and it's like dough. So trying to preemptively make sure I get 6 well-formed drops. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. I can also mention we have one tube here that we actually put the blood in about a half hour ago. This is a different version of tube 9 with the glycerol. We'll talk a little more about why we've created this, but we're going to be able to compare the blood that's been in glycerol for here over a half an hour versus the blood that we've just added to glycerol just now, which we're going to be spinning down presently. So we'll be able to see how the amount of time the blood cells have been sitting in the solution affects the results. So again, we actually have 2 versions of 9. And the next step is going to be, make sure these are kind of well mixed. If they don't look well mixed, you can just use like a clean, this is a clean pipette, and just kind of really make sure they're all mixed up well. And then obviously you're not going to use this for anything else anymore because it's going to cross-contaminate. So this is now plastic waste. Hopefully it doesn't poke the nose of a turtle in the ocean or something. All right, that is that. So next we will get a tour of the centrifuge.
All right.
All right, so I'm going to...
All right, so... yeah, so I put that turtle in there just because there's plastic waste is so nasty. In the medical, hospitals generate so much single-use plastics, which is necessary because that's how you keep things like from cross-contaminating and stuff. But in general, as much as possible, my public service message is, reduce single-use plastics and definitely don't, most people probably saw that video of the turtle where they had the straw in his nose. It was so sad. And that's just the tip of the iceberg. All right. So any questions about the setup? So you see, it's pretty straightforward. So what we're going to do now is the blood is in there mixed up. Something's happened. Now we're going to do that assay. We're going to spin them. Basically, it means makes gravity really, really strong in the tube. And then if the cells are intact, they'll get pulled down. And if they have exploded, the hemoglobin will stay in the supernatant. Let's have Tina tell us all about it. Here we go.
All right, so next we're going to centrifuge these test tubes. Unfortunately, we have more test tubes than we can finish in one round. But let's start off with how to use the centrifuge. So number one, make sure it's plugged in. Number two, on the back side here of the centrifuge, you have a switch. And so now it's on right there. And then once it's on, the display will light up. And it should already be set on the settings that you need. So we're going to spin these for 5 minutes and 5,000 RPM. So you might go, well, it's only saying 500, but if you look up here, it says times 10. So 500 times 10 is 5,000, so this is good to go. RPM means revolutions per minute. So that means that it's spinning 5,000 times per minute. That's quite amazing. So we're going to get a lot of G's, and it's going to separate out the components of this fluid based on density. And so we're going to get the more denser particles at the bottom. So our red blood cells will be down there forming a pellet, hopefully. All right, so next we're going to press open. And if we look inside, there are slots here that are numbered, but as David mentioned before, you should label the test tubes, because once again, we're gonna have to do this twice. And so just being organized from the beginning is kind of key. So next, I'm just gonna make sure once again that everything is balanced, because otherwise this thing will not be happy. And so
that's kind of, remember those sanity checks, right? Each stage, you know, to make sure you didn't mess up or your lab partner didn't mess up, they should be the exact same distance or the same height. And if they're not, then you know, like, oh, wait, something we got to fix. And again, things happen. That's why you need to have these little, in my world, we often call them dummy checks. Like, you know, did I do something, did I spaz here? I tend to not use that anymore in class, because one time someone came to the dean and said, he's calling us dummies. And it's like, no, I'm not calling anybody dummies. I call it myself, dummy check. Did I mess up here? But you can call it a sanity check, because that's like less loaded in the world of loaded words. So she's checking to make sure that I didn't mess up there and that it's the exact same height, so we're not going to be off balance. And I think in a previous take is where I mentioned make sure the numbers, we're going to have lots of things where there's lots of separate tubes with different numbers. So you definitely want to make sure you're labeling things well.
So I'm going to just stick things right across. So if I were to only do 2 tubes, now this would be balanced. But then I just keep adding tubes across from each other. As long as they are the same volume, they have the same weight, and everything should be fine. And I do not need to cover the test tubes with any parafilm or anything like that, because you can see that they're slanted, they're angled out, and we're going to have the centrifugal force moving everything out and downward. So there should not be any risk of anything spilling over. All right, so we're now set. We're going to have to save 2 test tubes for later. You would close it, and then all you need to do is hit start. And this thing should start up, and then it stops automatically after 5 minutes. So the centrifuge has just finished. I could hear it kind of slow down. And you can also look on the numbers that it's slowing down right now. It says 62, 58 RPM. And so you can really, see that it's slowing down. The lid probably won't open until it's completely still, because, you don't want to lose any fingers. There we go. It's also giving a nice little beep. So anyway, I'm just going to take these out. And since I have them numbered, I don't need to pay attention to exactly where they're at now. It's more important for me to get the next batch started. And so I can rearrange these later. So next, you guys would only have number 9 left, which is the glycerol. But remember that we're doing a little special experiment now where we left another glycerol in for a much longer time. And so it turns out I have to balance the centrifuge, but for you guys, you would only have this one. And so what you need to do is create a dummy tube that weighs the same. And so what I done here is I measured out 3 mLs of just water, and then I added 6 drops of water to make sure that they're exactly the same. And so you would just kind of put them right across from each other and do the whole process. So in this case, since I do have our special number 9, I'm going to stick it here, and they're right across each other, and we can start this centrifuge up one more time. And we'll come back in 5 minutes and take a look at that. Now I'm going to start taking a look at what's happened in my other tubes. So we have 1, 2, 3, 4, 5, 6, 7, and 8. And so if we just kind of do a visual look of these, you clearly see number 4 and 5, they have clear supernatant. So the fluid on top of the pellet is known as supernatant. And so those have clear. You also see it in test tube 7. And then all the other ones have some shades of pink. And you can see a little bit of a pellet down at the bottom here too. So we'll wait until we have the next batch too, so we can do our full comparison. We're gonna have to take a look at some of these pellets under the microscope to actually figure out if they crenated or if they're just the same as they were before. Because remember, they get pink when the cells actually lyses and we get the hemoglobin out there. But when they crenate or they stay the same as they were from the beginning, that's not going to be indicated by your supernatant. And so in those cases, we most certainly should break out the microscope and take a look at them and see if they're actually swelled up, or sorry, shrunken up, or are the same. All right, we'll come back in a few.
All right, so we finished spinning down the last samples here. Now we can look a little closer at what happened. You know, again, the one that was in distilled water, you know, basically osmolarity. All right, so at this point, I'm gonna run this through. Again, this is all something you can click on and watch, so you don't have to necessarily write this all down right now, or you can if you want. When you are recording your data for this kind of thing, you know, you do want to be able to say, yes, hemolysis occurred, you know, which is the first thing you'd get since it's a red supernatant. But in your lab notebook, under your observations, you'd also want to describe what it looks like, you know, red supernatant with no pellet. Like on your exam, on your lab exams, you know, you will see pictures of tubes. You know, in the real world, the old world, we used to actually see actual test tubes and stuff. But you have to know not just did hemolysis occur, but how did you know that it occurred? You know, so make sure when you are recording your data in your lab notebook that you describe what the tube looks like and why that's how you knew that hemolysis occurred, was because it was a red supernatant, and you know it was complete hemolysis because there's nothing left in the bottom as a pellet, right? So I'm going to go through this. I also have pictures of all of these tubes as well that you can look at after the, you know, so let's go, I'll continue with this, but know that you can go back and look at this. And also I have pictures that I put onto the module as well that have pictures of the tubes that you can look at. 0, this is the most hypotonic you can get. We can see the supernatant is bright red, and if you look for a pellet, it's really not a pellet, which is telling us that all the cells are popped. All we got are little shreds of membrane in there. And in fact, a little clue, when we look at this under the microscope and you're trying to see what happened, like, you know, imagine you were in a room full of balloons and then every balloon got popped. And then you walk in the room and like, let me see what it looks like in this room full of pop balloons. It's like,
it's not going to look like much of anything. Maybe you're going to see some shreds of rubber on the floor. So people get kind of frustrated when they're trying to like, what am I supposed to see here? You know, think about that. Again, we have varying hypotonic solutions. And one of the things that's interesting is we go to the 0.1 osmolar, 0.2 osmolar. And the 0.2, even though these are both hypotonic, we have red, which tells us we had lysis going on, hemolysis. Notice the pellets are actually different. Here, not all the cells necessarily popped. This test tube 3, this one with 0.2 osmolar was 0.1 molar NaCl. As we've done this lab over the years, we actually get different results, actually depending on the freshness of the blood. Blood that is fresher, the cells are healthier, more robust, they can actually take a slightly hypotonic solution and they're gonna puff out, but not necessarily pop. Whereas the older cells are not able to sustain that kind of increased pressure and are more likely to explode and create this hemolysis condition. So, what you get in this tube might actually depend on just how old or how healthy the blood cells are. And we can see the one that is in the 0.3 osmolar solution. This is what we would hopefully expect the cells to be in an isoosmotic, isotonic solution. And we see in general, the cells all just got pulled to the bottom intact. There is a slight blush here, which you wouldn't really expect, except I think in this case, the cells have been sitting around for a while. They might be just some cells died off just because they're getting old. Not necessarily because of the solution did it to them. This is the 0.6 osmolar solution, the really hypertonic solution. And here we see also the cells are intact, no cells exploded. But I don't know if you can see from your, can you see from your point of view there? Like I can see from my point of view, like a pretty.
Yeah, actually, you can see it pretty well. Which one of these do you think has crenated cells in it?
The one on the right.
The one on the right, you can definitely see the pellet is smaller. This would be like, the 100 raisins versus the 100 grapes. They're more shriveled up, so they compact down more. So here we actually can see, using just this kind of macroscopic view, that these are different, and this probably crenated versus this one. So just from looking at these, you actually can see this difference between the isotonic and the hypertonic saline, which is kind of cool.
Different size and pellets. When I was talking about the raisins, think about the grapes. These both have exactly 6 drops of blood, the exact same number of blood cells in them, but the amount of space that those blood cells are taking is different. And this is our first clue that these cells here probably crenated compared to these, because they're not taking up as much space for the same number of cells. And we're going to be able to follow up on that when we take them and look at them under the microscope. And obviously a crenated cell looks very different. It actually looks all shriveled up and spiky. You can see the cytoskeletal elements poking out against the membrane. They look kind of like, I don't know, pufferfish or something. All right, so test tube 6, this is the ammonium chloride. You can see we have no pellet. All the cells seem to be lysed, and the hemoglobin dissolved in the supernatant. This one's a tricky one. We'll talk a little more later about what happened here. This one's not so intuitive. It very likely did not follow your hypothesis, which again is all right. Hypothesis does not have to be correct. 7, this is going to be the 0.3 molar, which is also 0.3 osmolar sucrose. And we can see a really nice well-formed pellet with a relatively clear supernatant. Here we have the urea, and we can see the urea is also got a red supernatant and really no pellet. So we're assuming here most of the cells popped. And then finally we have the glycerol, and we have two different conditions here. We can start with this one here. This is the glycerol where the cells were only in for a few minutes before we spun them down. And we can see some of the cells lysed. There's red here. That means some of the hemoglobin escaped into the solution. But there's a pretty well-formed pellet, which is telling us a lot of the cells actually did not break apart and then got pulled down, complete down to the bottom of the tube. Whereas in this one, where the cells sat in the glycerol for about an hour before we actually spun them down, there's no pellet, which is telling us that the longer you sit in the glycerol, the more likely you are to explode. And we're going to talk about that more in detail as well. So kind of why that happened and why that makes sense. And actually we'll even talk about how this part would be different with human blood cells versus bovine blood cells. So we'll also provide pictures of each of these tubes as well for you, so you can refer to that as well when you're writing up your discussions about results and discussions about what happened here.
Okay.
I have a quick question about the test tubes. Did they say that the test tube 6 was the urea when it was labeled the sucrose in the lab?
Yeah, something got a little off for me too when listening to it.
So say that again.
I think test tube 6 and 7 were mixed up in the experiment that you guys taped. I think you said 7 was sucrose. In our lab, it says it should be urea.
Oh, interesting. Yeah, so follow what it might be we were I revised, I shifted around every once in a while to kind of keep, so if I did do that, then follow whatever I called it in the video.
Okay.
Yeah, so I would call it whatever I did it in the video. I will go back and double check that, and I might have to add. I don't know if hopefully you're all impressed. I've kind of figured out how to do these cool little flippy tags in Premiere and stuff, so that you guys can see in general what's going on. So in general, you can see we find the results are pretty much what we expect, except for what? Which one is kind of weird?
Glycerol.
What's that?
Glycerol.
Glycerol we said was going to penetrate, and it did. It penetrates and caused hemolysis. You know, it caused more or less hemolysis depending on how long it got to have a chance to penetrate and bring in water and explode things. So that one was kind of what we expected.
The ammonium chloride.
The ammonium chloride, that's the one that is weird. And when I first started doing this lab, I had no idea why the cells were exploding, and I couldn't find any good explanation of it anywhere. I would talk to the people in chemistry and try to get their sense of what's going on at the membrane. And it's only more recently that I've read some explanations of what's going on that actually makes sense. So I'll spend a few moments explaining what's going on just so you know. And I should mention, ammonium chloride actually is used it's used to preferentially lyse red blood cells, but not white blood cells. So any white blood cells were actually intact. They did not explode. Only red blood cells will explode with ammonium chloride. In order for this to make sense, I need to describe, we're gonna jump ahead a little bit. If this is an RBC, and so it's gonna, and let's say, and here's my plasma. One of the main ways that carbon dioxide is carried around in your blood, right? CO2, and you think of oxygen as bound into the hemoglobin, but CO2 is mainly goes like this. CO2 drifts into the cell. Inside the cell, there's a thing called carbonic anhydrase. CO2 plus H2O becomes carbonic acid, H2CO3. And then carbonic acid is a weak acid which dissociates into H plus bicarbonate, HCO3 minus. And then what happens is there is an antiport. And the HCO3 minus leaves the cell out this antiport, but to keep the charge balance, if I have a minus thing leaving, I need to have a minus thing coming in. So I have a chloride enters the cell. So this is, they call it the band 3 protein. It's basically a chloride bicarbonate antiport. And so now we have bicarbonate HCO3 minus. So this bicarbonate in the plasma is the main way that carbon dioxide is actually being transported in your blood. Like when we talk about this in more detail when we do the cardiovascular system and respiratory system later in the semester, we're going to revisit this. But the basic idea is this mechanism of turning carbon dioxide into carbonic acid into bicarbonate is just part of the way your body carries around, transports CO2. But this antiport here is going to be the reason why your cells are dead. And it's what I saw Morgan sent this thing out about, you know, weak acids, weak bases. So basically, H2CO3. So my NH4Cl becomes Cl minus and NH4 plus. But then this NH4 plus actually is a weak acid that breaks apart into NH3 plus H plus. And the NH3 actually can diffuse into the cell. But then once the NH3 diffuses into the cell, it's now a weak base that comes back apart and becomes NH4 minus again plus OH. And then those OHs combine with the H pluses that are happening here as the carbonic acid is dissociating. And as that happens, it's going to drive this reaction this way. And these bicarbonates are going to leave, more Cls are going to come in. So basically, we are getting ammonium chloride entering the cell through this really circuitous process. We're ending up with ammonium chloride in the cell because of all of this machinery going on. So the reality is this ammonium chloride is coming into the cell, and so the water follows and the cell explodes. But it's only in RBCs. This antiport does not exist in white blood cells, so you don't have this, so this whole thing doesn't happen. So this only lyses RBCs. And that's because only RBCs have this antiport that's necessary for this whole process to happen.
For our write-up, what should we say about this?
Don't worry about the details of it. you can say, like, because of a particular membrane protein involved in exchanging bicarbonate with chloride ions, ultimately the ammonium chloride does enter the cell and therefore upsets the osmotic balance. So this is why it's okay if it wasn't intuitive to you in your hypothesis, like, oh, for sure. I mean, obviously it's going to explode because of this. All right. So any comments or questions about this part? All right, let's take a 10-minute break. At noon, we'll come back and just finish up with the last part of today's lab. And that'll give you guys a chance to just kind of absorb some of this. Before I do that though, I will show you, share screen. Hold on, let me turn this off. Share screen. Where is it? Where is it? There it is. Okay, so now if you go beyond these, here are some of these just so you can see. And what I realized I should edit these a little bit, so I will take these, and they're here right now just so you can see them, but I'm going to bring them into Photoshop and I'll crop them and make them a little prettier and label them better. So you have these as well to take a look where you can kind of check out, and I'll give you the actual files as well. I'll give you links to the JPEGs and stuff. I need to, so these are here for you to look at. If you want to print things out and put them in your lab book, that's not a bad idea, to do some of the appropriate pictures so you can really tell what's going on here. And like I said, I will make these prettier over, like, what time? Yeah, before like mid-afternoon, these will be prettier. So you can come back. I would not print these out yet. They're going to be prettier later this afternoon. But let's take a little break, and then when we come back, we'll just wrap things up here. Okay. So there's just a last couple of things here. One is the microscopy, like to really definitively tell if a cell is crenated or not. You look at it under the microscope. Obviously, we're not going to do that. I did make a little video kind of showing the technique for making a wet mount. Like, one of the things when you're making a wet mount of blood or lots of different things, you don't want to just, put it on a glass and put a cover slip, because then the water starts evaporating out the sides, and pretty soon, things are all dried out. So you want to be able to kind of seal it off really well. So you could just have it sit around for a while and it's not going to keep changing on you. So the way you do that is with kind of Vaseline and making this little gasket around the edges. So, so just to kind of show you what that process is, and then just kind of show you some images of what the blood cells typically look like, and again, so things that you can describe, you should describe these pictures in your lab notebook, even if you're not going to actually see them. You should describe at least what does a photomicrograph of a crenated cell look like compared to a normal cell, for instance. So we're going to check this out. So again, I'm going to make those prettier soon, but now it's going to be a little thing showing how to do a wet mount of the red blood cells. Here we go. All right, so if I want to actually see if my cell's truly created, the only way to really see that is to look at them under the microscope. I'm going to talk a little bit about how to prepare a microscope slide to look at the blood. You know, so let's say this is test tube 5 where I'm assuming they created. First thing you're going to need to do is resuspend the blood cells up in the solution, obviously. You don't want to totally mix it back up. If you put 2 concentrated blood cells on a slide, it's hard to see any single blood cell. It's like trying, if you're interested in looking at a grain of sand, but you're looking at a whole handful of sand, all you're going to see is just crazy sand grains everywhere.
Yeah.
And a few of them. So don't get too ambitious about resuspending them. You just kind of you back up in the solution there, and then you can grab them up, but you really want it to be pretty darn dilute, or else it's going to be really hard to see any individual cells. And then we want to actually create the actual slide. All right, so to make a wet mount of the blood to see what happened to it, we want to make it so the cover slip is sealed, so the water doesn't evaporate from the solution if it just sits there. And so there's a nice little technique to make a sealed wet mount using Vaseline. You start, this is just regular old petroleum jelly, Vaseline. You take a bit and you make a thin layer across this webbing between your thumb and your forefinger. You know, it should be thin but present. Then I am now going to take my cover slip, and this is just very thin glass, and I'm always going to make sure I hold it just by the edges because I don't want to get my fingerprints all over it. And I'm going to, for each of the 4 sides, I'm going to put the side into the Vaseline and just drag it. Maybe it's like about 1/4 of an inch, and it brings, I don't know if you can see that, there is a Vaseline has created a seal on that one side. And then for 90 degrees, go again. Now I have Vaseline on another side. Rotate it again 90 degrees. And Vaseline on 3 of the 4 sides. Again, 90 degrees. And I'm running out there. That's good. So here you have Vaseline on all 4 sides, kind of underneath, and it's going to make a nice gasket. When I put this down on top of my main slide, it's going to seal around the edges, and I can show you how that works right now. So here is my slide. I'm going to take a drop of my resuspended blood cells here. That's probably plenty. I'm going to take my slide and start one side, drop it down. I'm going to use, the back of my fingernail. I have to push it down a little bit. And what we have now is a sealed wet mount. The blood suspended in solution is sealed inside the Vaseline underneath there. So this can be sitting around now for like a half an hour, and it's not going to change. If you did not do, it's a bit of a production to actually do this sealing, but if you didn't, like 10 minutes from now, you start having like crystals probably under there as the water evaporates and leaves into the room. So this allows you to have a nice slide that you can come back to and show other people as well. And this is what you would do for a variety. we talked about in the lab, and it'll look at test tube 1, look at test tube 3, look at test tube 4, look at test tube 5. So you do the same process for each of those, and compare and draw. For our online version, we'll show you pictures. But this is the only way you'd really know what really happened to the blood cells, particularly if they didn't lyse. So the whatnot of the blood.
Okay. So that is the basic way of making the wet mount. And then on the next module here, the next part, well, let's just submit. Where did I put it? Here, blood photomicrographs. I just basically, these are just things I found on the web. And these are just ways for you to get a sense. Normal RBCs, which of the test tubes would be isotonic?
I think it's, let me check.
Number 4.
4.
Okay, so number 4, which other one would be isotonic?
6.
Yeah, so the sucrose as well. So a red blood cell on its own looks kind of like a little pucker. It's round, but there's no nucleus in it. So it's kind of flat with its sides kind of sucked in. So normal red blood cells, they're red, but they don't look that red because there's so little hemoglobin. They're very thin and they're shined with a bunch of light. So they'll look kind of reddish, but they're not going to look that red under, imagine you have a very thin layer of kind of reddish fluid, but the thinner the layer is, the less you're really going to see it being super red looking. This is crenated. Which one would be like the crenated version?
Number 5.
Test tube 5.
Like test tube 5, exactly. So the idea in your lab notebook, when you are kind of writing this part up, I would probably either describe or even print out some of these. Again, I'm gonna put links as well on these two actual images that you can take. But which of your test tubes is this most likely from? This is going to most likely be like this, the 0.6 osmolar, whereas this is going to be 0.3 osmolar. This is kind of showing the same thing. The blood cells, typically they look kind of like this because they're round with that kind of pucker. They tend to look, this is what a normal healthy blood cell looks like. But when they crenate, they turn into these little asterisks, or I was calling them pufferfish. They always get all spiky looking with all the cytoskeletal elements kind of starting to jab out and stuff. This next one on the bottom, this is kind of showing isotonic, kind of normal round cells with a little pucker. Hypertonic, where they are coming in and getting to be these little pufferfish. Hypotonic. What can you tell me about the nature of this hypotonic solution? Is this, do you think could this be test tube 1, the distilled water?
No, the difference is the osmolarities was not that much.
Exactly. So what is the only one of our tubes that this is possibly?
Number 7.
No, 7, that was the sucrose was just, that would be completely isotonic.
I'm sorry, 7 on ours was urea.
No, urea is going to be, what happened to the cells in urea?
They lysed.
They lysed. Here we have cells that are intact. They're just kind of bloated looking.
Maybe number
3,
3 if they're healthy new cells.
Exactly. So these are the ones that are slightly hypotonic. They've taken up water, but they haven't exploded. These would be like the ones, you know, when we saw in test tube 3 that there were still cells in the pellet that hadn't lysed. You know, they are not lysed, but they're definitely going to be kind of fat because they've taken on water and they're inflated. So this would be like slightly hypotonic, right?
Which test tube would this be for?
It would be like the
3.
1 molar NaCl, right? The one that is, it's hypotonic, but pretty close to 0.3 osmolar. Right? Does that make sense?
Would glycerol fall into that one as well, or no?
I would say, I don't know. That one's, because that's really not hitting a really nice equilibrium. This is at least an equilibrium. I would say don't worry about the glycerol. Glycerol, I don't know. If the glycerol has, some of the glycerol has made it, some of them, where they are depends on how long they've been sitting. So I would say you don't know. The glycerol is, you don't know.
Gotcha, thank you.
Let me continue with this. Here are these images, again, kind of showing cells that are basically looking pretty happy, cells that are crenated, and cells that are slightly hypotonic. Again, slightly hypotonic meaning they've inflated, but they haven't inflated too much, because what would this look like if this was test tube 1 or test tube 2 or the urea or the ammonium chloride? What would I see in the microscope?
You wouldn't see cells. They would be little fragments.
Yeah, I wouldn't see much of anything. If anything, I would see maybe little bits of schmutz floating around. But in general, you know, the cells have exploded and shredded. You know, there's not really much to see at all. Other thing, in this picture, this thing here, this bigger thing, again, we're not gonna, in anatomy, you probably remember, white blood cells are larger than red blood cells. That's a white blood cell. Like, so if we used ammonium chloride, this dude would still be alive. Everything else here would have popped. But the white blood cell, which does not have that ammonium chloride, I mean, that bicarbonate chloride transporter, would have survived the onslaught. So, you know, for your lab notebook, you know, I would say you should connect, like when it says what do you see under the microscope for tube 1, for tube 3, for tube 4, for tube 5, you know, try to describe what it looks like. I do want you to actually, like on an exam, on your lab practical, I would feel okay showing you a picture of blood cells, and you should be able to, you should be able to look at these two and tell me which one was in the 0.6 osmolar versus a 0.3 osmolar.
Okay.
I do want you to be able to recognize, like, you know, here, you have bloated cells, shriveled cells, and kind of normal looking cells. I do want you to have the sense that those 3 states exist and what kind of osmolarity would lead to those 3 situations. So that makes sense?
Mm-hmm.
Okay. So the very last thing, this is ironic, but one of the things that's always frustrating to me is you're looking at something under a microscope and we're talking about it swelling and shriveling, but it's all kind of this little thing you're looking at under a microscope. So in order to give people a more hands-on feel of bloated or shriveled or isotonic or hypertonic or you know, we take sausages of dialysis membrane, that same stuff we used for the osmometer. And we fill it with, they all have the exact same fluid inside. They're all filled with 0.3 molar, 1.5 molar sucrose. And then we put those sausages, which are big, they're about the size of a little hot dog, into different solutions. We put one into distilled water, one into an isotonic solution, one into a hypotonic solution, one into a hypertonic solution. We put one in this really sugary solution, and then let them sit there and let you poke at them with a glass rod so you can really feel what is it like if you've got something sitting in a hypotonic versus a hypertonic versus isotonic solution. You know, so you have a hands-on feel rather than just have to like do it virtually. All right, so we're gonna watch, you're now gonna watch Tina poke at little sausages with a glass rod. So here we go. All right, the final part of today's lab.
All right, so we're learning about osmosis and tonicity in this course, and so here we're going to do a demonstration of osmosis. So what I have here are 4 kind of sausages made by dialysis tubing. So we fill them with 1.5 molar sucrose solution. So all 4 have the same, and they're about the same size. Next, we're going to stick them into different solutions, and we're going to see which way the water is going to move. As you remember, osmosis, we're going to see the water is going to move from a low solute concentration to a high solute concentration. Or another catchy way to remember that is that water chases solutes. So I'm going to stick each of these in here to distilled water. I'm going to put one in here to 1.5 molar sucrose solution, which is the same as the inside of the sausage. Remember, all of the sausages have 1.5 molar sucrose solution. And then this last one has 3.0 molar sucrose solution. If I stir it, you can see it's quite, concentrated and sticky and thick versus here it's still has sugar in it, but it's a lot more dilute. And then distilled water, of course, is just distilled water. I'm also going to leave this control sausage here because when I come back in 30 minutes, I can tap on this and kind of feel what was the original kind of, consistency of this sausage, and then compare what has happened to the other 3 sausages. So we'll come back in 30 minutes and take a look at them. Hey, Mama. All right, so we're back at our sausages here made out of dialysis tube, and it's been actually an hour. And so just to kind of check on these, here's our control. So you can see it's, kind of this consistency here. I can lift it a little bit of bending, but, we filled it with sucrose solution. This has been in distilled water, and if I push on it and also lift it, you can see it's very rigid. So this one has taken in water and got quite taut and rigid. This one has been in the same concentration as we put inside the tube, and so this one is very much like the control sausage, kind of same consistency. And then if we go to the one that's been put in a more concentrated solution, you can see that it's pretty soft and flaccid. So obviously this sausage has lost water to the outside solution. All right. Now it's up to you to figure out exactly what happened, which way did water move, and what do we call the solution that we put them into.
All righty. So the basic idea there's a little chart in your lab manual that just asks you which one's hypo, hyper, isotonic, which way is the water moving. And again, it's just another part of trying to really get you to think about osmosis and really think about the terminology and really think about what's happening, because it's going to be at the core of things, in physiology from, in all, again, the very last couple of weeks of this class, when we get into the, the kidneys and stuff, it's all about osmosis again. So you really need to understand these concepts. And so that's why we're trying to play them out in lots of different ways, with osmometers, with blood cells, with little sausages. So hopefully, as you keep seeing it, you'll start feeling more and more kind of get an intuitive understanding of it. And just, I kind of feel like, just in case you're doing this at home, that really ooey gooey stuff, we don't actually use 3 molar sucrose. That's actually use some corn syrup in there to really give it a gooiness. So it really feels it is super sugary, but to make it feel more visceral, like, yeah, it's really concentrated sugar, which is what we want you to really understand. We added some of the corn syrup in there to make it feel really sugary and gooey, just so no doubt in your mind, there's a lot of solute outside of the sausage there. So just in case you're wondering, wow, is 3 molar sucrose really that gooey? It's like, no, not really. So any questions about all of this? So on the web page, I included a link to upload your write-up when you're done, but that's not for a couple of weeks. You should definitely work on it, and while it's fresh, it's you don't have to wait that long, but it's not due like tonight or something. I have been grading. I've got some of the chem review labs graded, but it's trickier. That I'm trying to figure out the best way to do it. It's grading through Canvas and trying to make comments and I'm still trying to find the best way. So I apologize that I haven't just graded all of those, because I'm still trying to find the best method. So I'm just so used to using a pen and circling things and putting arrows, and it's not intuitive exactly how I'm going to efficiently grade these things yet. So I will be getting to them, and I am getting to them. But yeah, if you haven't, make sure you've been finishing up your jumping lab. Make sure you're finishing up the osmometer lab as well, because if you fall behind, it's just going to get, you'll start digging yourself into a hole. So the more you can just have it off your plate, that means you can go do a virtual Zoom beer with a friend rather than have to sit and work on a lab that night or something. So there is no pre-lab for Thursday. Thursday, we're gonna kind of shift gears for a day and kind of get more into more of the process of, I talked about literature searching and how is information kind of shared with the rest of the scientific world, and how are you, efficiently access that, and we'll introduce that in more detail. But there's no pre-lab or anything for Thursday, so you can spend your time just kind of writing this one up.